I'm not the biggest fan. I feel like my dad whole life.
All right, is everybody ready to get started for today? Yeah? All right. So today we're going to sort of continue our conversation about synaptic transmission. Remember last class we finished off talking about the electrical basis for neurotransmission, essentially action potentials. We talked about them getting integrated at the axon hillock, that connection from the axon to the soma of the neuron's body, and being propagated down that axon by way of saltatory conduction through the sodium channels, particularly that comprise those node of Ranvier sections on the axon, which are those areas that have densely packed sodium, voltage-gated sodium channels in between those chunks of myelin. Everybody remember all that? And then that action potential got transmitted down the axon through to the presynaptic terminal, and then it basically resulted in neurotransmission. Okay, so that was a lot of the electrical basis of neurotransmission. Is when we have these action potentials, were they occurring in graded format? No. What was graded? I'm sorry? What was graded? Speak up, please. I can't hear. What was graded? Yeah, there were graded potentials in the dendrites. But the action potential itself was all or nothing phenomenon. Remember? So a couple of key features. We said action potentials were all or nothing. We said they were unidirectional. What do we mean by unidirectional? Huh? Yeah, they only move in one direction, down from the hillock towards the axon terminals, okay? And we finished off with that nice rendition of the synaptic cleft here from Bobby Voltage. Hopefully you guys all remember him. They're obviously, I don't know if I told you guys this, they're from Stanford's biology program a while back, and they make a bunch of other videos and stuff like that about science. Why can you tell that they're from Stanford? Because what? Huh? Why? Yeah, Cal Sax, they're big rivals. Okay, they're all in the Bay Area there. It's a big rivalry between Berkeley and Stanford. So it's nice to have some fun with neuroscience and do all that kind of stuff. In semesters past, we've had projects for the class where people made their own versions of videos like this. It was a lot of fun. And if you're interested, shoot me an email. I'll send you some clips that you can see of what other people have done in the past. Try to have some fun with it. So let's see here. So where do you think we should start for today's class? If we're going to talk about we're going to talk about the chemical basis of neurotransmission. So neurotransmission ultimately is both electrical and chemical, okay, which is actually part of the lyrics for the song, if you guys went back and revisited it. And there's an ionic basis for it as well. So if it were you, where would you start today's content discussion? If you were Drake, where would you start? Where? At the bottom, yeah. Okay, so here we have Drake. Okay? Now, what he's showing us here is apparently he has a very keen interest in science, and he's decided to do a little example for us about the sodium-potassium pump, setting that ionic basis for the action potential, okay, or our resting membrane potential. What we're looking at here on the left-hand side is our extracellular space, and this is our cytosolic fluid here in the intracellular space. Now, what's Drake doing here, outside of looking like a fool? So, what's in red? Na. And what's in purple? And what is he doing other than looking like a fool? Yeah, doing what that sodium-potassium pump is supposed to be doing, exchanging 3 sodiums and 2 potassiums across that cell membrane, okay? So A little bit of fun for you, okay? You guys don't have to look so bored, okay? This might be trust me, it's going to get more boring. We're not going to have as much Drake going on the rest of the class. We're going to get into some real science, but maybe that's more exciting. So let's just jump right into it. We'll talk a little bit about how some of this electrical basis is forming the chemical foundation for neurotransmission, okay? So we're going to start thinking about neurons as information processing units. Okay, so what we mean by that is basically these neurons can be conceptualized as integrating a bunch of synaptic inputs. So we talk about these things in examples in class and all that kind of stuff with very, very simplified models. A presynaptic neuron and a postsynaptic neuron. But outside of some very rare situations, most of the types of processing that's happening is integrating thousands, but really truly millions, if not billions of neurons firing together all at once, or sometimes not together. And slightly not at once, okay? And somehow, these little biological units and the soma they're in have to integrate all this information that's coming in and somehow make a decision about how to relay that information. And it's happening on incredibly fast millisecond temporal resolutions. So we'll talk about that a little bit about that and how that's happening. And I want to start thinking about neurons within that context. Everybody on board with that framework? Okay. So everybody should remember this idea that, of course, our postsynaptic neuron is going to fire this action potential if there's a depolarization that happens. So we talked about action potential, right? We're graphing it out as resting at a negative resting membrane potential at about what level? Yeah, negative 50, 60, maybe a little down to negative 75. Let's just say negative 65 conversation. So it's resting, hanging out like Simba, stalking Zazu, nice and low, right? And a very negative resting membrane potential. Something happens, it gets some influx of positive charge, gets a gradient potential. So it rises up a little bit because there's a little bit of influx of positive sodium ions coming in from the extracellular space. And then if it's enough to exceed a threshold, we have that characteristic action potential, the overshoot, and it comes back to normal. We said the rising phase of the action potential was due to voltage-gated sodium channels, and the falling phase of the action potential was due to voltage-gated potassium channels. Okay, you guys should have that stuff down. Okay, know it like the back of your hand. Know those basic elements of it. Okay, and we'll talk about moving forward how these different types of electrical signals can start to be integrated at the synapse through chemical means and to operate through neurotransmitters. So when you see this abbreviation of NT, it's referring to neurotransmitters. They're different chemicals that are packaged within what we're going to call synaptic vesicles in the presynaptic bouton and then travel across that synapse, or the space between, as Dave Matthews Band would refer to it as, and somehow result in this ionic influx in the second postsynaptic cell, okay? So it's going from electrical to chemical and then back to electrical again as we're transitioning between neurons in this chain of communication, okay? So remember this idea that we had our EPSPs and IPSPs. So EPSPs were excitatory postsynaptic potentials. So this would be a postsynaptic potential here. Okay, we had that graded potential, okay? And on the other hand, it could have been a negative hyperpolarization, not hyper, and it could have gone more negative if it was a chloride ion that had influx, right? Okay, that would have been an inhibitory postsynaptic potential, or IPSP. So be familiar with these types of abbreviations and know the chemicals and the ions that can be involved in leading to both EPSPs, IPSPs, that is GABA, and chloride ions, glutamate, things like that, okay? Glutamate for excitatory postsynaptic potentials, perhaps influxing with some sodium ions. We have the chloride ions leading to inhibitory postsynaptic potentials, okay? Now, we're talking about this in the context of communication between cells. Right, and of course, not just between cells, but between neurons and brain cells, but of course, peripheral neurons as well. And very rarely do they receive just these singular inputs, like we mentioned a few moments ago, in these isolated, just one presynaptic, one postsynaptic cell situations. Of course, they're kind of like us. We're getting bombarded with a lot of information all the time. Okay, we have friends that want to go out to this bar, and then friends that want to go out to this party at a different place at the same time. We're getting all these inputs. Then people want to go to study groups. Then other people want to go, to the gym and work out. And everybody wants to do something else. You're getting all these different competing inputs. You have to make a decision and fire, so to speak, and go and do whatever it is you decide to do. How do you adjudicate all that competing inputs? Okay, we'll talk about some of the ways that these different inputs, both excitatory and inhibitory, can influence a cell and how it can lead to both action potentials being fired and continuing that signal, or reducing the probability of an action potential and quieting down that potential signaling. Okay? So, again, we can sort of see this classical action potential exhibited. Of course, it started by this excitatory postsynaptic potential that exceeds threshold. So for those of you guys who don't like suffering through my horrible artwork, we can see generally the same thing represented by a much better artist here. Okay? But of course, we can also see that inhibitory postsynaptic potential that's a little bit more hyperpolarizing. Remember, hyperpolarized, which means it's more to the extremes away from the central point of, say, 0. Okay? So it's hyper, so it's going down here. And then we can see this excitatory input there. Okay, so that's just a way to illustrate that. Now, we talked about integrating these signals from multiple different cells. Okay, so here we can see our traditional exemplar soma, or body of the neuron. And what we see is it's getting a lot of axonal inputs onto it from a bunch of different cells. And the plus and minuses here represent excitatory and inhibitory influences. Okay, think of it you can think of it in all sorts of different ways, however suits your fancy. Think of it as the cartoons with the little angel on your shoulder and the little devil on the shoulder. One person saying, go and do this, and the other one's like, no, I don't know, you shouldn't do that. This, that, and the other. Excitation and inhibition. Okay, one is leading to a greater likelihood or probability of that cell firing, and the other is leading to a reduced probability of that cell firing. Okay, so somehow this cell is getting competing inputs. Don't fire, don't fire. Yeah, sure, go ahead, fire. Yeah, go ahead, fire. Ah, what could it hurt? Why don't you fire? Okay, so basically, the long and short of it is you can think of it as it sort of works in an algebraic basic sum. Okay, whatever competing inputs end up having more, whatever there ends up being more of these competing inputs will generally tend to be the winner, so to speak. All right, there were caucuses last night in Iowa, so it was like whoever won, I guess, had more people go to their side of the room or something like that, right? It's a sort of an algebraic. You just add up, is there more signal on this side or more signal here? Okay, it's the same idea here. In this case, you're getting greater excitatory influences than just the two negative ones. And we can see if we sort of measure that here with this electrode at the axon hillock, it seems to be exceeding that threshold. We get this extra positivity, and we can see that action potential likely being fired down that axon. So let's take a look at some of the ways that this can work as well. So we can see that working in inhibitory fashions too. So if you have too much inhibition now you have 3 inhibitory, well, I should say enough inhibitory influences where it can override our 3 positives. So what we're looking at here is a difference between a lot more positivity. See how there's red sort of shown here, but you don't really have a lot of the negative coming in. But look what happens when you see this extra patch of blue, sort of representing the influx of those chloride ions coming in. Now they sort of start to algebraically overweigh some of the positive influxes that's being shown here by the red, in which case it's not enough to sort of exceed that threshold. And if we measure here, we don't see anything being sent down that axon. Okay, so it's just a very basic conceptual framework here of understanding some of these pretty simple ideas. All right, so there's a couple different ways that these signals can be integrated, okay, because it gets both the red and the blue, so to speak. Okay, so it gets these influx of negative ions, the chloride ones, and the positive ones of the sodium, and somehow they basically add up and give that integrated signal. Okay, and there's a couple different ways that we'll talk about that. The 2 big ones that are important to know for this class are temporal summation and spatial summation. We introduced this idea briefly last class. All right, so the idea with spatial summation is that we can measure this cell, and we can measure whether it exceeds whether the potential of the cell exceeds our threshold for firing an action potential based upon the different inputs. Okay, so here we're talking about spatial summation. So what we mean is if you have these small little graded potentials, each in their own right is not enough to exceed our threshold for firing. What you need is to sum them. Remember we talked about that algebraic summing idea just a few moments ago? We need to add them up. But they have to happen, or one way that they can be added up in a way that's meaningful and will lead the cell to fire is if they happen in a very spatially close area. Okay, so basically inputs can come from all sorts of different areas of the cell, and those ions actually have to sort of travel to create an electrostatic sum. Okay, and if the overall sum of those EPSPs are sufficient to depolarize that cell here at the hillock, it'll work. But we have to have basic spatial summation, so they're coming in from a reasonable amount of the same area. Okay, and we can compare that to temporal summation. Okay, so what we're looking at back here they're all coming in relatively sort of on the same area. Okay, but now we're not talking about space so much, we're talking about time. Okay, so if they come in this close enough period of time, bam, 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 you know something's probably important happening because it's all happening at once. So maybe that's a signal you should pay attention to and relay to your friend down the chain of command. Does that make sense? You can kind of think of this stuff as sort of chain of command issues or communication. If you're getting input from multiple sites that are all very close to you and a couple people all right next to you, that probably means that there's something systematic happening. So maybe you should pay attention to it and relay that signal to your partner. Same idea, maybe that's not coming for 3 or 4 people real close to you, but maybe just a single one, and you keep getting text messages every second or two, bam, 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 it's blowing up. You're like, okay, maybe something's happening, I should pay attention. Okay, so the difference between spatial summation of signals versus a temporal summation of signals. We understand how each can operate, okay, and they do, and sometimes together. Okay, so the closer that they arrive together, the more likely they can add up to exceed that threshold. Because if they're not coming in fast enough together, if they're just real slow, you're going to get that little positive influx of sodium ions coming in, but then they're going to disperse across the rest of the intracellular fluid, and they're not going to sort of stay together to keep that measurement together, that extra positive charge, to open up that voltage-gated lock where that threshold is. Is that clear? Yeah. All right. So, spatial and temporal summation, okay? If you're interested in more of the details, one way to study about this is to make yourself a table. Here, the publisher has already made you a simple sort of table about the type of electrical signals that are happening, right? We have action potentials, EPSPs, IPSPs, okay? Tables are sort of boring to read. They're boring to put on slides. They're boring for me to present. But they're really helpful for you to create on your own. So that's one of the reasons I have this here, is to make a suggestion that you make your own. Even if it's just literally replicating and copying down the same flipping table. It'll just help to organize your thoughts and organize your understanding and studying of each of these different parameters. And as we move forward, that's another suggestion that I make as we start talking about more of the anatomy and the neural systems in the weeks to come, is one really helpful study tip is to spend a couple bucks and get some tracing paper and some colored pencils and trace over the figures, trace over the anatomical connections. All right, something that was recommended to me when I was an undergrad student, and I found it really helpful to study, and I know neurosurgeons who still do it to keep up and stay fresh on certain things. Speaking of which, get her up. And so, anyways, so let's talk about a couple other aspects as we move in a little bit more depth about moving towards our chemical basis of neurotransmission, because we can sort of summarize what we've already brought to the table about the electrical idea. Okay, so know our IPSPs, know our EPSPs, know how they summate both temporally and spatially, okay, and how this basically operates to integrate both our excitatory and inhibitory connections. And of course, it's going to lead to whether or not a cell ends up producing or not producing an action potential. Okay, and you can think about this as representing that perpetual tug of war, as I talk about it here, but those competing requests that we all have to adjudicate about. Where do we want to go at night? This friend wants to go study. This friend wants to go out and party. This friend wants to go out and watch the Mavericks game. This friend wants to go down on Sunday and I don't know, watch Tony Romo lose football games or something like that. So, right, we have to resolve these different competing inputs. And of course, sometimes we're torn between two options, and the neuron is too. And these are some of the ways that it adjudicates that. Okay, sorry for Cowboys fans, guys. And really, I'm sorry for Cowboys fans. I'm a Bears guy, so I gotta have a little bit of fun. All right, so electrical neurotransmission is one essential way that neurons communicate. But that electrical signal goes to the end of the neuron. Do you guys remember the neuron doctrine? What was the neuron doctrine? I thought you guys said you remember the neuron doctrine. It was what? I can't hear you. All right, who gave us the neuron doctrine that we attribute it to? Yeah, Santiago Ramon y Cajal was one of the big proponents of it. Of course, other people contributed to those ideas and things like that at the time, but he largely gets a lot of the credit. So Ramon Santiago y Cajal is not this guy, but what did Santiago Ramon y Cajal tell us that we consider the neuron doctrine? Yeah. Cells are individual, and they're separate from each other, right? Which doesn't sound revolutionary to us now because you guys are all on top of this already. But why was that a big deal at the time? What was that in contrast to? Golgi, right? Not the apparatus, the guy. And what was his contention? The net, the reticular theory, right? So we have this neuron doctrine that was essential in leading forward from that alternative hypothesis or model. And our neuron doctrine said that these are individual discrete units. Well, here's the thing. These electrical signals cannot jump from one neuron to another. Right? The electricity just sort of stays within it. Somehow that signal, though, is transmitted from one neuron to another. All right? The way that that happens is chemical, but we didn't always know it. Okay? So one of the ways that we knew that was from some really critical advances that was given to us by Otto Lowy. That's who this gentleman is here, okay? So he was basically studying some aspects of neurotransmission, and he did a really famous study, and we'll talk about it a little bit. So basically, he was trying to figure out how neurotransmission worked, and he had a really cool idea. He woke up in the middle of the night. He's like, yes, I got it. And what do you think he did? Huh? You think he experimented with it? No, he passed out. It was in the middle of the night. He was tired. He went back to bed. He's like, so what happened the next morning? Hey, he totally forgot it. He woke up the next night. He had the idea. He's like, I'm not going back to bed. He ran to his lab, cooked up this experiment. Okay, or at least maybe he wrote it down. I'm paraphrasing to the big large picture here. So anyways, what did he do? So what he wanted to do was try to understand how communication might be happening between cells. So what he was able to do was identify that there was a chemical messenger that was being sent that was serving electrical transmission. Okay? So if you measure basically heartbeats, or in this case, the force of contraction of the heart, okay, what you can do is you can stimulate a vagus nerve that goes to the heart. The heart is a muscle and it beats, and when that happens, he was able to isolate it in sort of this representative beaker. And the idea was that when you do that and you stimulate with our vagus nerve, okay, you can measure both the first heart and then you can measure a second heart that's in its own solution or bath that's connected by a tube to the solution from the first one. Okay? Now you've only electrically stimulated one. Okay? And you measure that beating of that heart, okay? And then as you measure that, look, beforehand they're both beating at basically the same rate, the same way, okay? Now, what happens? You stimulate the vagus nerve, okay? And you measure both of them again. Now, here's the thing. The stimulation of that vagus nerve led to a change in the electrical measurements. But moreover, the really cool finding was not only did it lead to a difference or a change in the electrical measurements of the first part, which is kind of like, duh, right? You did something to it, so it makes sense that maybe something would change. But then there's this other heart that he didn't do jack to. No, he didn't even do anything. It was just sitting there hanging out. It was just connected by a tube of some fluid. But what happened was this heart started exhibiting the same pattern of changed firing, both amplitude and rate. What does that tell you? Or what did it tell him? Any ideas? Come on, pretend you're in the laboratory here. You got your white coat on.
Signal rather than just an electric one.
Yeah, so there has to be something more than electricity happening here. There has to be something chemically that's moving through the fluid that was presumably released by virtue of the electrical stimulation on the first heart. There's some chemical that must have been released and transferred from the fluid and somehow affected this heart. Does that logic make sense? You guys all on board? Can you put your phones away up there? Thanks. And so the idea here is there's another part. What other part here is helpful towards arriving at that conclusion? I'm sorry? I can't hear you. The fluid? Okay. What about it?
There wasn't any electricity, or there was electricity probably running through it?
Well, we didn't have electricity. Let's say there wasn't electricity running through it, per se. Let's take a look at the time difference. So this heart changes, but really, we said also this heart changed too. That was cool. But there's a difference in how it changes as well. So we can double down on this and look at a second component. Look at when this changes, nearly immediately. Look at when this changes. Is it nearly immediately? No. This heart is chilling the heck out. This one is not. It takes some time for it to start winding down over here to get to the point that this was at much sooner. Again, that shows us a lag, a temporal lag. So it's going to take some time. The chemicals have to move. You guys follow? Okay, so we have those two multiple converging lines of evidence there. All right, so we can talk a little bit more now about this, the chemical basis of neurotransmission. Now that we've identified that there is some chemical basis, let's ask, what is it and how the heck does it work? All right, so we know the axon is traveling down our axon, hits our axon terminal, somehow releases in somehow results in something that releases chemically that somehow affects the downstream part in the communication chain. Okay, now we're going to talk about a key role that calcium plays. Remember we introduced the ion calcium earlier? We said it wasn't really involved so much in setting the resting memory potential the way that sodium and potassium was. We already talked a little bit about chloride coming in and forming those IPSPs. All right, but we haven't really talked a lot about calcium, but it plays a really critical role, but not so much in the soma as much as it plays in the axon terminals. We'll take a little bit of a look at how that's happening. And remember I said that we have those chemicals, our neurotransmitters like serotonin, norepinephrine, dopamine, these types of neurotransmitters, these chemicals that are packaged within synaptic vesicles. So we'll look at that. We'll look at this in a minute. But if you just think of, let's say, a vesicle, just a big circle of stuff. Okay, that would be a vesicle in our synapse, and our neurotransmitters would be chemicals localized within that. Okay, so that's just to provide some scope and context of the different types of things that we'll talk about. But you'll see better pictures than my crappy drawing. Okay, I know it's crappy. All right, so let's take a little bit of a closer look. Okay, what we're looking at here is a presynaptic cell's membrane. Okay, so here's a little cartoon conceptualization of these vesicles like I drew down here. So at the surface of our membrane, there's a lot of these vesicles, okay, and they contain little neurotransmitters, and we can start to see, so these are electron micrographs here, we can see the sort of membranes of our cell before they're stimulated and after they're stimulated. We see a lot more of these bumps, these vesicles starting to make these indentations. So what it says is that when you stimulate a cell, our membranes at the end of them start to take more of these little vesicles towards the ends of the membrane. We can see that in these pictures. Okay? You guys see the difference between this one and this one? Okay, that's all you got to take away from here. We'll get progressively more and more detail. All right, so let's take a look at some of the factors of what's happening here. Okay, so we can start with our presynaptic neuron in our postsynaptic dendritic spine here. Okay, we have these again are our synaptic vesicles, and within them are those little neurotransmitters, just like I drew them. Okay, except better looking. All right, so they're hanging out. We have this electrical stimulation that comes in, okay, and we start to have these electrical action potentials come down, and these synaptic vesicles containing our neurotransmitters are going to fuse with the membrane. So this little circle here is going to fuse with the membrane and release those chemicals into the space between the synapses. All right, and then those neurotransmitter chemicals are going to bind to receptors that we have on this postsynaptic membrane and start inducing a cascade of events that lead to communicating that signal to the second side. Okay, so when that happens, we can see a couple different things. First of all, there happens to be an increase of calcium when that happens. Okay, in fact, it will not happen without an increase of calcium. So as that greater positivity happens, there's calcium channels on this membrane here, and just like we said, those voltage-gated channels for sodium on the dendrites are really important to unlocking that voltage-gated gate to be open to allow the sodium ions to rush in. We have voltage-gated calcium channels in our axon terminal boutons here. When there's that positive action potential that rushes in, okay, there's those voltage gates on the calcium channels open, and allows those positive charges to rush in from calcium. And calcium is going to start getting to work, and we'll show you exactly what they do. All right? So this is our basic framework of what's going to happen. All right? Now, when these things happen, we'll see that calcium plays this really critical role in allowing our synaptic vesicles, these suckers, to fuse into the membrane and release those chemical neurotransmitters. Okay, so when we talk about that process, that's what we call exocytosis. Okay, so that neurotransmitter is being exocytosed into the synaptic cleft. Okay, so here's an example of, well, if we look down here, we can see a bunch of our little synaptic vesicles, like I drew them here. Okay, so we can all draw circles. Like I suck at drawing, but I can draw a circle. That's all they got here. It's just a circle. I got more detail. I got neurotransmitters in here. They don't even put them there. Okay, but the idea is the same, and they're doing this fusion process. They're coming down along the membrane, and they're fusing. And this way they can release that transmitter across the cleft. And it goes through a cycle. We're going to talk about that cycle. Now what we're looking at on top is an actual representation of one of these synaptic vesicles. Okay, and all this stuff on it are a bunch of different proteins. Okay, so you guys got a pen and a pencil? Start taking that. No, I'm kidding. You don't have to remember all this. Okay, they're just there to show you how complex this actually is, and to let you know that we're going to distill it down to just one or two key things, and hopefully capture your interest so that you can follow up with some classes that get into all the fun nitty-gritty biochemical science of all these other proteins that are involved. Okay. Yes, sir.
Is that like two scales, like the vesicle is actually that size compared to all the proteins around it?
Roughly. Yeah, I mean, it's representative. Yes. All right, so let's talk about some ways that this happens. Okay, so we're going to zoom in, and we're going to simplify it. So instead of looking at all this stuff, SNAP29, SNAP5, SIC3, SNAP-tag, VAM4, synapsin, da-da-da-da-da-da-da. All these boring abbreviations, it's just totally overwhelming. Okay, you don't have to know that stuff. Just want to show it there to introduce the level of complexity that's really at play. We're just going to keep it really simple here. Now I'm going to introduce one or two key players. Okay, so this corner yellow strip is like representing, whoa, the wrong one. There we go. Okay, that's like representing this. You see how they're both kind of in the same shape? So it's just a bottom piece of a synaptic vesicle. And just like our membrane that we talked about, it also has a phospholipid bilayer. And that's why it looks basically like a phospholipid bilayer. Now it's got a couple of these proteins wrapped in it, within that membrane, and they extend outside of it, and a couple other ones. And then this is our presynaptic membrane. Now outside of it here is the synapse, okay? So this is all intracellular fluid, and here we have that presynaptic membrane, and again, it's phospholipid bilayer too. So let's take a look at some of the players involved, okay? So what I want to introduce you to are just a couple proteins to think about, okay? We have synaptotagmin, okay, this orange protein, and we have SNAP25, which is this green protein, okay? Now in red, that wraps around over here, we have syntaxin, okay, so we just got 3 or 4 of these guys. There's another one here, synaptobrevitin in blue. But 4 things, 4 colors, and we also got these little purple dots. What they're representing is calcium. So remember we said calcium comes in when the action potential rushes down into the axon terminal gluton, opens those voltage gated calcium channels, calcium rushes in. So now we've got all these positively charged calcium ions. Here's what happens. What we want to do is exocytose. Okay? Right now it's not exocytosing. You've got all these chemicals in here that want to come out and part. Alright? They've got an invitation. They're waiting outside the bar. They just got to be let in. Here's what needs to happen. This membrane needs to fuse with this one and create that exocytosis that we introduced a moment ago. We'll talk about exactly how that happens. Okay? And we can boil it down into simpler terms. So now we're looking at the same proteins, similar colors, similar names. They're just not wrapped up in these little curly fries. Now they're just a little bit straighter cartoons. Everybody on board? So we're also just still talking about the same thing. We're just keeping it nice and simple. Yeah? Alright, so here's what we got. We have our presynaptic terminal. It's got all these green little neurotransmitters. Calcium comes in because right now they can't bind. Okay? What happens from our first point is now the vesicle comes in and these proteins start to wind together like ropes. They start tethering and tying. Kind of like you're tying off a boat. Alright? So if any of you guys like to do boating or anything like that, go party up by Sneaky Pete's in Lake Louisville. Okay, you think of tying off a boat up there. Yeah? Everybody go to Party Cove up there? Yeah, it's kind of fun. Oh, you had a question? You have Party Cove?
No, I was talking about Lake Louisville.
Yeah, it's kind of fun, right? Yeah, so you tie off a boat on the dock and it brings it together. That's what we're calling it docking. Just like you're tying off on a dock. Okay, so they start to tie up here. Now what happens is these vesicles are still not touching the membrane. Alright, they need to kiss together. How that happens is this. As they tighten, they complex and they pull together. As calcium rushes in from these voltage-gated calcium channel, extracellular calcium comes inside the cell. Okay, and then from inside the cell, it does what we call it complexes, and it binds with synaptotagmin, these little orange, little bubbly things, okay? And when it binds there, it induces a conformational change. It basically provides this mechanism to wrap all these proteins together. In our third step, it brings it real close. So now the boat is really docked, right next to the dock. And here, the boat actually, in our example here, becomes a part of the dock, which you definitely don't want to happen. Because you don't want your boat to become a part of the dock, because that means you have a hole in the boat. Holes in boats are bad. Here it's okay. Have I confused you guys? A little too much boat analogies? Everybody good? Okay. So, exocytosis, because of calcium-mediated, or we'll just call it calcium-mediated exocytosis. Okay? Or you might call it kiss and run, because they kiss the membrane and then they pinch off and they run back in, just like we saw on those earlier slides. Okay? So, we can talk about a couple other facets that happen during the synaptic release. Okay? When that neurotransmitter is released into the synaptic cleft, it doesn't stay there forever. So it has to get broken down or cleaned up because the biological systems like to be clean. We're always making messes, but we always require ATP and energy because our cellular systems are always cleaning up our messes. okay? So we'll talk about a couple ways that this is happening. Okay, namely, we'll talk about some enzymatic degradation, okay? And we'll also talk about what we'll call autoreceptors that can be important in managing release, okay? So our basic framework and model here is we have a presynaptic glucon, we have our action potential coming down, a positive influx of charge, opens up these voltage-gated calcium channels, calcium rushes in, it binds to our synaptic vesicles through synaptotagmin, right? It complexes, ties up along the membrane in exocytosis, our neurotransmitters releasing the cleft. Everybody on board? Yes, sir.
Does the vesicle, is it actually docked on the end of the gluton, just sitting there waiting for calcium? It's just floating around waiting for the calcium to come in?
Yeah, I mean, you can kind of see, representatively, I mean, they're kind of all over in the gluton. Sometimes they might be a little further away, other times they might be real close. There's a lot of them, right? So, I mean, by virtue of them, there being more than one, by definition, they have to be in many places, right? So, yeah. Sometimes you can imagine some of them are much closer, and some of them might travel a little bit. And this is what it's showing too. So there is a cycle. Remember, it says kiss and run. You can think of it's kissing the membrane here, and then it's running back and coming in, packaging up the mold. Okay, so there's a lot of recycling here. Synaptic vesicle recycling. So, anyways, we have our chemicals being released into the cleft, but like I said, they don't last there permanently. And there's a couple ways that these chemicals and neurotransmitters are cleaned up. Okay, we can talk one, which is an autoreceptor. So we can see that sort of representatively up here. It has little detectors on the outside that can detect a relative amount of neurotransmitter there, and it can basically induce a series of changes that will cease the release of those neurotransmitters. And we can also have reuptake pumps, which are little pumps, just like Drake's sodium potassium pump, where it pumps stuff back across the membrane. So you can have reuptake pumps on the terminal gluton here that will reuptake some of those chemicals and bring it back in and allow them to be repackaged into vesicles that can be recycled. Okay? So, any guys over here are SSRIs? Selective serotonin reuptake inhibitor. All right? There are pharmacological treatments for clinical situations like depression. That's one of the ways that they work, is they selectively, the first S, find a serotonin receptor, the second S, okay? And it's a reuptake pump. So it would be reuptaking the serotonin, but it's inhibiting it. So what that does is allow serotonin to last and to work a little longer in the synaptic cleft. The reason that seems to be efficacious for depression is because depression seems to be associated with deficits in operational serotonergic function. So it allows it to work a little bit longer by blocking its reuptake. You see all the logic and all that? Cool. All right, so we have those things happening too. All right, and okay. So here we have our postsynaptic cell. Now it's receiving these neurotransmitters, and these neurotransmitters are binding to receptors that hang out on this postsynaptic receptor. Now those postsynaptic receptors are just like the postsynaptic receptors that were on our presynaptic cell that initiated that action potential. So now everything starts, the party starts all over again. Okay? And now when they bind to this postsynaptic cell's receptors, it permits ions from the extracellular space here, such as sodium or chloride, to rush in and give an influx of charge. And the party starts over. Okay, so that's our basic idea. We mentioned the idea that there could be reuptake pumps. There's also the idea that these signals and these chemicals can be degraded. So sometimes those chemicals are released into the synaptic cleft, and instead of being taken back up through a pump, they can be degraded. So a good example of this is the neurotransmitter acetylcholine. It works a lot in, say, neuromuscular junctions and things like that, which is the junction between neurons and muscles. And it oftentimes can be broken down by an enzyme. So that's what enzymes do. They do a lot of breaking down of stuff at times. And in this case, it's breaking down acetylcholine. And the enzyme that does that is called acetylcholinesterase. So usually when there's an esterase or something like that. It means it's breaking that first part down. In this case, it's acetylcholine, which is the first part. So it's breaking down acetylcholine in the synaptic cleft. Okay? All right, so we already talked about reuptake and some of these transporters and how they that might work through some of the autoreceptors that can detect the relative amount of neurotransmitter in the cleft and send those signals back to the presynaptic cell, sort of self-regulation. It's another way of saying autodetector or autoreceptor. Okay? All right, and so that's our basic chemical synapse in that particular regard. And that is the basic for, remember we said most of this is unidirectional in terms of the action potential, right? And most of the time the sort of the canonical dogma here, right, the rule is that these chemical transmissions are mirroring that principle from the electrical transmission of unidirectionality. However, there can be a couple exceptions to this rule. One notable exception is a special type of synapse that can occur sometimes where there's actually retrograde signal. Okay, so if we think of our, let's say, our presynaptic terminal there and our postsynaptic dendrite here, okay, this is our synapse. Usually we have this unidirectional neurotransmitter being released to our postsynaptic cell's receptor, okay, and we have our sodium channel ions or whatever rushing through there, and that's unidirectional, pre, post. But you can also have situations where there can sometimes be what we call retrograde signal, okay, so it's kind of like a hipster likes to go retro, okay, what does it mean to go retro? Yeah, going back in time. Like the Huey Lewis song. Okay, going back in time. Going back to the future, friends. Okay, so it's kind of like that. Going back to. So it's going to go from this postsynaptic cell back to our presynaptic cell. So one way that happens is through nitric oxide. That's one of the transmitters. Basically, it uses gaseous transmission in that regard. Carbon monoxide, nitric oxide are two examples of ways that postsynaptic cells can send information backwards across the synapse to send information to the presynaptic cell to modulate transmission. Okay? So know one of those examples. So this is sort of one of the exceptions to the rule. Okay? But otherwise it typically operates from that pre to postsynaptic fashion. Any questions about this stuff right now? Okay. Another exception to the rule. Okay? We're going to call this Golgi's revenge. All right. Remember we started off talking about the neuron doctrine? Santiago Ramon y Cajal, neurons are individualized discrete units and they signal and operate in such a fashion. But fortunately the Nobel Committee was either clairvoyant or remarkably lucky in that they still awarded the prize to each of these competing colleagues of Golgi and Santiago Ramon y Cajal. And our friend Emil Golgi kind of got the last laugh post hoc, as we might say, or after his time had passed. So it turns out that we have some things called electrical synapses, or what you might call gap junctions. Okay? So the idea here is that an action potential can actually send an electrical signal directly from one cell to another. How do you think that could happen?
The membranes actually physically touching?
Yeah, so there's some special situations where the membranes of a pre- and postsynaptic cell are actually connected by virtue of some ion channel or some protein channels that allow that electrical signal to pass across immediately. And you can measure that electrically and compare that to the speed and the timing of postsynaptic potentials in that situation compared to times when you know that they're separate. And you can see a lag. Remember we talked about that lag in the Otto Loewy's experiment? Otto Loewy got the Nobel Prize for that too, by the way. I think that's our 5th Nobel in this class that we're talking about. So we'll talk a lot about these guys for obvious reasons. They made fundamental contributions to our understanding of the nervous system. So what the idea is that you can have a presynaptic neuron and a postsynaptic neuron basically tethered together by virtue of They're protein channels, and that allows that electrical signal to flow directly through those large channels and into the cells. And you can measure it with nearly no time delay. Okay, let's take a little look at what we mean. Here's a basic example. We have a presynaptic cell shown in yellow, and our postsynaptic cell shown in blue. Okay, what are we looking at? These pink things are our ion channels, which are comprised of proteins. Now, we don't draw them as proteins all the time because we like these really smooth, simple cartoons that look like kind of bubbles. But in reality, they're proteins, and they look like those curly fries that we saw in some of those earlier slides. Okay? So we have these protein channels, and they're on our presynaptic cell, but they're also on our postsynaptic cell. But they're so close together in these specific situations that they actually complex and they form together. And if they're both open like this, and they're together, it allows an action potential that's coming down from this presynaptic cell here to flow right through to the second cell. So you have direct, immediate, fast electrical communication. That's called a gap junction. Okay? And so in this way, the neurons are kind of connected sort of as you might think of as the reticular formation. And so it ended up being a little bit of both. And so we can see that beyond a cartoon. So here we have our cartoon, blue and yellow. Okay, so yellow is presynaptic, blue is postsynaptic. Here we have that same illustration. Okay, we have a presynaptic and postsynaptic, and these are sort of the gap junctions, and they sort of connect almost directly across. Okay, questions? Yes, ma'am. Well, I don't know that they even have a gate to be blocked. I think just the nature of those. So she asked what causes those channels to be open. That's a really good question. But remember that before we even started talking about voltage gated doors or gates on these ion channels, before we even got there, we were first talking about passively open channels that just allow stuff to move back and forth without a gate. Remember we talked about passive sodium channels, passive potassium channels in particular? So not all channels have to always have a gate. And so that might be sort of what's at play here. Okay, and that leads us to sort of the next thing I wanted to talk about is these different types of gating in these channels. So we talked in last class about this idea of a lock and key mechanism as an example of neurotransmitters operating as a key to unlock the gate that is the door on our respective ion channels. Okay, so we have our lock here. Of course, she's not very happy at the key, and the key is very sad here because he's getting rejected because she says he's just not her type. Very sad. All right, can't do this next week for Valentine's Day. That just wouldn't go over well. Or the week subsequent, I guess, is what that is. But anyways, so we have this idea of a lock and key mechanism, and certain locks will only open when there's certain keys that fit just the right way. And ion channels are very similar to that when we talk about some of these basic aspects of postsynaptic neurotransmission. Okay, so we'll introduce a couple terms here. When we talk about ligands, or ligands, whatever you want to call them, all right, the basic idea here is that they're chemicals that can basically operate as that key to unlock certain receptors on the postsynaptic cell. All right, so what we're looking at here is a representative receptor on our postsynaptic cell. Okay, and so we have our intracellular compartment here, our extracellular compartment, and this is that ion channel. Now remember, we said this is a nice, comfortable cartoon representation, but they're really comprised of those curly fries of proteins. Okay, and there's different subunits of those different sections of the protein as they wind back and forth across the membrane, and different subunits, as we call them, have different spots that our neurotransmitters or chemicals can bind to. Remember, out of low we showed us that there was chemical neurotransmission. Something's happening chemically. We have that chemical being released from the synaptic vesicle, traveling across the synaptic cleft and binding to our postsynaptic receptor. Okay, so that's basically what's referred to as a ligand. Now, if it's an ion channel that's usually blocked and then is open, not when there's a voltage gated change, but when there is a chemical binding change, you might call it a ligand gated channel, because it doesn't give a darn flying hoot about the electrical charge. It won't open it. What it opens to, the key that unlocks this door, is a ligand. Whatever that ligand might be, a particular neurotransmitter. Some receptors only care about serotonin. All right, they tell dopamine to go screw itself. Other receptors only care about glutamate. Don't care what acetylcholine has to say in the conversation. Okay, either way, they're all considered ligands. Okay, so the idea is that it can operate in this capacity, and we can see that evidence just broadly speaking. You can have ligands that work in more of an endogenous way, some of our hormones and neurotransmitters that we talked about, dopamine, serotonin, norepinephrine, glutamate, GABA, all these types of things. There can also be exogenous ligands or other chemicals that are not endogenous to our own internal production, but which are introduced as outside chemicals that can hijack these systems and work on them too. All right? Sometimes they're medicines and pharmaceutical drugs. We mentioned SSRIs. Other times they might be more illicit drugs like cocaine that hijack these systems too. Okay? So anyways, that's the idea of our ligands, forming the basis of our chemical neurotransmission. We introduced the idea of acetylcholine a little while ago. We said it operates oftentimes at the neuromuscular junction. Okay? And so acetylcholine basically combined to two different types of channels that are ligand gated for it. There can be a nicotinic receptor and there's a muscarinic receptor. We'll take a little bit closer look at that. Now, some of these channels can or chemicals like acetylcholine can fit really nicely on the cholinergic receptors. So cholinergic, we just mean receptors for acetylcholine. So remember we said a moment ago that there can be exogenous ligands that can hijack a normal receptor. Well, we can see some evidence of that as well in nature. So you can have a cholinergic acetylcholine receptor where you can have other chemicals come in and block it and operate it. Okay, so if you have a neuromuscular junction that's operating by virtue of acetylcholine, you have these cholinergic receptors that's allowing a muscle to work when a nerve sends an impulse to it to contract or relax, but it's blocked by an exogenous ligand such as the poison curare, okay, or bungarotoxin is a poison from snakes. Okay, what they do is they block our nicotinic acetylcholine receptor. So here we have our acetylcholine receptor, okay, and it usually responds to acetylcholine or black arrow, but you could have something like curare which comes in and it blocks it. Okay, so what happens there is we can have a situation where these receptors are blocked and they're no longer responsive. And so if they're at the neuromuscular junction that allows the person or the animal to move and they don't work, guess what the person or the animal does? Nothing. They can't move, they're paralyzed. So that's why some of the Amazonian Indians would tip their arrows in this poison curare. It worked on the acetylcholine receptors to poison their prey. So don't kill them. But you don't need to kill it. You just need to stop it. And then you go catch it and then you kill it. And you have a delicious meal. Yeah. And it's not just us. The snakes did it through the bungarotoxin, alpha bungarotoxin that we talked about a moment ago. Okay? So we can see some examples of this operating in nature and how these systems are working normally, but then also how they can be taken advantage of and hijacked as well. Let's see here. Another couple points to mention about these postsynaptic side of neurotransmission. So we talked about this idea that we can have these postsynaptic receptors. Okay? We have these receptors here. Now, they can also be changed. They're not permanent. You can have things that happen based on experience, based on the inputs that it received beforehand. You can have a series of biochemical cascades of molecular and biochemical effects that are happening in the postsynaptic cell that can either upregulate or downregulate these receptors. What do we mean by that? You can add more receptors into the membrane to make that cell more responsive to that particular neurotransmitter, or you can downregulate it. Okay? So let's say you have a case of drug abuse and it's bombarding these postsynaptic cells with a ton of chemicals and drugs that it's not used to. Well, it has a natural mechanism to pull some of these and downregulate, pull some of these channels out of the membrane so that the cell becomes less responsive. Because it doesn't want to freak out every time there's this huge, massive influx of this particular drug. Right? The system likes to stay healthy. You know, it has a way of attenuating. Yes, ma'am?
Is that what happens with heroin, where they say like you get so high the first time, and after that you can never get high anymore?
Yes, you can have different forms of tolerance served by, or sensitivity, served by both upregulation and downregulation of receptors. Yeah, there's a lot of different ways that sensitization and tolerance works through upregulation and downregulation of receptors for all the different types of drugs. Yeah, that's how a lot of people are thinking about it and found it to operate. So, a couple key points to know there. We've got upregulation and downregulation. Now, a couple ways that these receptors can work. Remember, we're talking about this postsynaptic side of the cell now. Okay? They can work in what we call an ionotropic way or a metabotropic way. So, if the receptor opens from its closed state, remember most of these ion channels are normally closed. Let's say it opens. If it does that, in response to the binding of an ion, we very simply call that an ionotropic receptor. Okay? Okay, so we can also have metabotropic receptors. Now, what do we mean by that? These are receptors that open after a sort of metabolic process, so they require a second messenger system to be used. Okay, let's talk about that in a little bit more depth. Okay, so when we talk about our ionotropic receptors, this is a representative schematic one. You have a basic receptor, an ion comes in, or a neurotransmitter comes in, okay, it binds, it induces a conformational change that opens that channel, and the ions can flow through. Okay, very simply, ionotropic. Okay, it happens very quick because there doesn't have to be any other crap that happens in between. On the other hand, we can look at situations that we call metabotropic receptors that happen a little bit slower because it needs a couple of intermediary biochemical steps to happen. Okay? And so what it basically operates as is you have these messengers that are basically second-hand downstream messengers from our first messenger is, of course, the neurotransmitter. Our second one is the downstream one. Let's call it a G protein for purposes of conversation here. We won't talk about the details of what that is. Just know that there could be a G protein coupled receptor that induces a couple of metabolic reactions that allow that ion channel to open up a little bit slower. Okay, so here's what we mean by that. Let's take a little bit closer look. This is an easier way to look at it. Here we have our receptor. If a ligand binds to it, it doesn't give a flying hoot. It's not an ionotropic receptor. It doesn't respond to that ligand. However, if a ligand binds to this other receptor, so here we have that ligand in the neurotransmitter, it binds, okay? Now this cell, this receptor, is coupled to a G protein. Okay, don't worry about all the details of what that is, just know it's a G protein. Okay? When the ionotropic receptor works here, it binds to the ion together with the G protein and activates a series of effects through the second messenger that then comes over and unlocks this door from the inside. Okay? When this happens, now it'll open that channel to allow the ions to come through. Alright? It's kind of like these ions have a fake ID and they're trying to get in the club, and the bouncer out here is like, no dice, you're not getting in. So they go and they talk to their friend who's a bar back at the place, like, dude, come on, let me in. The bar back goes, runs, talks to the bartender. Bartender goes, talks to the bouncer, goes out to the guy and says, all right, fine, let him in. Come in through the back door here. Opens it up from the inside. Really? I used to be a bouncer, so it used to happen all the time. Yeah, I once carded Bon Jovi. I didn't believe it was him. And so I made him show some ID, seriously. He lives on the. I lived in Jersey at the time. He's a Jersey guy, and he was down on the Jersey Shore, and he looked like every other idiot on the Jersey Shore. Cut off shirt, holy jeans, cowboy hat, which is like what everybody looks like in the summer, really. And everybody's like, yes, Bon Jovi, that's Bon Jovi. I'm like, no, it's not. Go back to work. They wouldn't keep working, so everybody's whispering. I'm looking, and I'm like, all right, go back to work. I'll figure this out. I go up to them and was like, hey, you know, I'm head of security. Like, are you Bon Jovi? Yes?
Sorry to interrupt you, Bon Jovi. I have a question about that. Have you heard of those studies that the ADH meds are now causing Parkinson's? Like, if it's long.
I have not heard of those studies.
Okay.
But what happened with Bon Jovi was that.
Demographic, I don't think they know who that is. So.
Anybody know who Bon Jovi is?
Yes.
Okay, we got a pretty good demographic. And anyways, he's like, yeah, I'm him. And I didn't believe him. I said, show me some ID. So let me see some ID. And so he showed me. I was like, all right, cool, let's get some beer. We got him beer, and then people found out it was him. And he got mobbed. He left in 2 minutes. So, anyways, no, I don't know about the study of ADHD linked to things like this. But if you show it to me, I'm happy to chat about it.
Well, no, I was going to ask about how the receptors and all that would work. If you're talking drug-related,
Yeah.
heroin, and all.
Well, nobody really knows how the ADHD works. So right off the bat, it's a tricky question. It's a tricky question to a tricky answer, or flip that around. It's a tricky answer to a tricky question. So we don't really have a really clear mechanism of action of understanding how that works. We don't know how schizophrenia, although there was a study that just came out, I think last week, that's supposed to have unlocked some of those questions. We don't really know what the mechanism of action is in autism. There's a lot of things that we don't understand just yet, similarly with cancer. We're trying to get there. We've cured some cancers. It's how science works. We're getting there. That's a situation where it might be the case, but it hasn't been fully adjudicated and resolved yet. But I haven't seen that study. If you shoot it over to me, I'm happy to chat about it, and we'll see if we can sort it out. So this idea of metabotropic receptor, kind of like, again, you've got not allowed in with the vague ID. They go through the back door of the bar, the bouncer lets them in, and everything's cool. They can come in. So no ionotropic receptors, no metabotropic receptors, know the respective speeds of each they're in. Okay? Here's another way to look at it in a little bit more complexity, but the idea is identical. A basic ionotropic receptor, it is closed. Okay? You don't have to go home, but you can't stay here. All right? Ligand comes in, show the real ID, come on into the bar. We got plenty of booze to buy. Spend all your money. Make sense? Okay. Here, of course, it tries to bind. It doesn't work. It has to go through another second messenger to get put on the guest list. They're like, dude, that's my friend. Let him in. Okay, fine. This is our second messenger, which in this case is a G protein coupled receptor. All right, it sends off a series of biochemical cascades, these intracellular messengers. It binds here intracellularly, opens up that postsynaptic receptor, allows influx of ions. Metabotropic. Cool? Everybody good? Awesome. Let's finish off talking about a couple other things here. We can now start to build up from this framework. Okay, so we've talked about this very simple model of one neuron to neuron communication, and we talked about how, of course, that's just a simplified model, and there's actually a lot more happening, and we've already had some discussions about the role of convergence of multiple inputs here and how the cell soma is involved in integrating those signals. We talked about temporal summation, spatial summation, and factors like that. Okay, I just wanted to sort of introduce this idea that we can look and start to understand how these neural systems are operating, both through what we call convergence and divergence. Okay, so what we're still looking at here in sort of this basic system is just a serial network, not Captain Crunch, but serial as in S-E-R-I-A-L, not the other one. Okay, and I spelled that right, didn't I? Yes. Okay, just want to make sure. You never know sometimes, you know? Well, I never know at least. You know, we used to be able to spell so much better. I don't know if there's any people from like my like Stone Age generation, we used to have to write with things like pens and pencils on pieces of paper, and we got tested in cursive writing and stuff in elementary school. It was brutal. I failed. I didn't fail. I got C's in handwriting, but that was like failing at the time. And now we write in computers and we get spell check automatically and autocorrect. It's awesome. But I don't know if you guys find yourself. I always find myself questioning some of the spelling sometimes because you're so used to having it fixed for you. Anyways, we have a serial line of communication. Okay. Now, basic series of connection. Now, this case is representative of the visual system, so we're talking about some cells in the retina. All right. And it's just one to another, to another, to another. Very simple, basic chain of events. Okay? And indeed, the nervous system can and does operate in that type of an organizational fashion. All right? But we'll talk about these ideas of convergence and divergence. Okay? So let's look in a little bit more complexity about how things are actually operating. And as we look at this, keep in mind everything that we've talked about for the last couple days. All these electrical patterns of firing and spikes, graded potentials, inhibitory ones, excitatory ones, action potentials, all these chemicals bouncing back and forth across synapses, retrograde signaling, all this stuff flying back and forth on millisecond timescales with all these ions, all these chemicals, all these neurotransmitters, vesicles fusing, non-fusing, calcium-needed stuff. All this, everything is happening as literally ions, sodium and potassium, flipping back and forth through these channels, through electrostatic repulsion and attraction, electrochemical gradients, okay? But scale this up into all the connections that are made. Not this serial line, but these ones where now you have everything converging, okay? So you have multiple cells represented here in green. They're all converging onto this one, which then integrates the signals from these onto another one. And this cell integrates the signals received from these two neurons, which are already integrating signals from 6 others. So we have this idea of convergence. Okay? Now we can also then look at how that expands to the idea of divergence. All right? So you have a single neuron then that gets all of this and then projects it forward, and then, just like many other things, it starts to spread. So the neuron that it projects to then can project to a little more. Okay, so you have all this chemical electrical stuff happening, ionic concentrations and driving forces and electrochemical forces all at once at building into the scalable network. For me, it's kind of overwhelming and kind of like awe-inspiring that we're even reasonable beings and not this haphazard random pattern of firings. Like there's a systematic organization to this, right? Like we're conscious and human, we do things on purpose and we're not just random and sporadic. Like this is really elegant when you start to think about the electrical patterns that have to happen to underlie most of the operations of our nervous system. Okay? For me it's very impressive that this can be accomplished, and a lot of people are making a lot of headway. They're trying to understand how that's happened, how that is happening by virtue of modeling this with like computational models and things like that. So if any of you guys are interested in aspects of that, computational neuroscience is a very, very burgeoning field. And so we'll start to take some snapshots as we move forward in the next couple classes and talk about some of the methods and the ethics of research that scientists use to understand these broad level patterns of electrical activity as they converge and diverge, okay, from both we've already talked about some of these methods with single cell recordings for our graded and electrical potentials, or action potentials. We'll even look at this at EEG that measures these electrical patterns of activity at the scalp, representing millions and billions of these cells firing at once in cohesion, and all the different levels in between of measurements. And we'll talk about some of the ethics that are involved in guiding that research, some of my most favorite topics. And we'll talk about some cool stuff. Hit me up if there's questions. And a point of reminding, what happens, so we have a class Thursday, right? And then we have a class Tuesday, right? No. Is it a day off? No, there's a test. Where? The testing center. Make sure you register. That information is on the syllabus. Get a seat. show up on time, rock it. Okay, there will be review sessions, use all the resources available, all that kind of good stuff. Okay? Thank you for your attention. Hi.
Okay, I'm going to start off this conversation by telling you I don't do drugs.
Okay, that's okay.
I am allergic to
I'm not going to judge
Texas.
you. You're allergic to Texas?
I just moved here, like, a month ago.
Didn't you hear God bless Texas?
Yeah, no, do you see these eyes? Holy shit, my eyes have been red for a week.
You know, I get so allergic, like I sneeze on all the different allergy seasons. It's brutal. Like, I don't know if it's pollen or there's something in the air here. So
It's worse
I know
here.
what you mean.
And I broke out in random hives for 3 days in a row, like just half my face
You know,
hives.
it's not you. It's a common, like, people like have to adjust.
I look like I'm high. I don't do drugs. It really upsets me.
You know, the
because
thought
people
never
just
crossed-
judge
It
me.
never crossed my mind, so there's no need to even.
Okay, well, I'm glad you're the only person. I may be going to a funeral,
Okay.
and
I'm sorry
they
to
don't
hear
know
that.
if it's okay. It's a family member, whatever.
I'm sorry.
They don't know if it's gonna be Saturday or Monday, and it's not in Texas. It's in Louisiana. So what should I do about the test? Because I don't know what.
When would you like to take it?
I can do it like when I get back, like Wednesday maybe. I don't know when I'm coming back.
I'm happy to work with you with whatever you need. Okay. Just tell me when you would like to take it, and we'll schedule a time when you can go there and just take it.
Okay. Awesome.
But what I need you to do is email me so I know your name and ID number so that I can make an exception specifically for you. Okay. So I can go in electronically and like basically cook all this stuff up and structure it such to accommodate that.
Okay.
But I need to know I need to know ID number, I need to know name, and that stuff that I'm not going to remember now if you just tell me.
Yep.
Because I have no memory.
Okay, that's totally fine.
Just shoot me an email.
Okay.
And we're good.
All right, thank you so much.
You're welcome.
I emailed you last week about maybe changing the test date for me, and then I sent an email earlier today about because I had an interview next week.
Oh yeah.
I forwarded an email to you.
Okay.
Would that be enough?
I mean, I haven't seen it, but
Okay.
presumably it should be fine.
Okay.
Yeah.
And so how would we go about changing the date?
Same thing I just said to that lady as well, which was, I don't know if you heard or not.
Send an email.
I have an email of your name. Presumably your ID is your net ID number too, so it's probably in your email. Tell me a time that you would like to retake it. And in email, by the way, so that it's like on record
hard copy.
and I can
Alright.
like go get it. And I'll just coordinate it and I'll set it up, but I need to know a time to reschedule.
Okay.
A day and a time that you are free. And just email it to me, and we'll be good.
Okay. Is he going to be either Monday or Friday? Is there a better time for you, or does it matter? Probably Monday. I can do whichever.
When are you going to your interview?
It's going to be Tuesday to Thursday.
Where's that?
It's in New York. Yeah. So we'll be flying
Good
over
luck.
there. Thank you.
I could do it Monday.
Okay.
But I'm gonna need and not to be insulting or anything like that, but I just need to cover my bases. That if you take it Monday, I need to have assurances that obviously
I'm not going to share.
the ETD honor code is
for sure.
expected to be abided by and followed. And so if it's found out not to be, which is going to be pretty evident because I know what the distributions of test performances look like.
Yeah.
And so when that starts to get skewed, it's really easy to identify. So I mean you can do it
Monday.
Monday.
Okay. All right.
Again, please
Yeah,
email
absolutely.
that to me so I know the actual.
Yeah, I'll send another email with the date and everything in my name. And
Awesome.
okay. All right. Thanks.
Thank you. Hey man.
I was wondering if I know a lot of why questions don't really have a lot of answers, but do you know of an evolutionary advantage of why there would be both chemical messaging and electrical? So we talked about the gap junctions, right? For ions are able to pass right on through, and also chemical messengers, which are a little bit slower. So is there reasons why we have both and not just one or the other?
Well, it's a really good question. I get a little pedantic when I answer that. What I mean by that is like, I try to answer it in the very most accurate way that I can.
And sure, yeah.
The way that the question was worded was, do I know the reason? Now, nobody on Earth knows the reason because nobody was here when it was created.
I guess is there a functional difference between the two?
Yeah, so what we can say, and what you're asking, I think, is there an evident advantage?
Right, yeah.
Right.
That's what I was thinking is an evolutionary advantage.
I don't mean to get pedantic, but it's like, it is kind of my job to make sure people...
No, absolutely. Is there an evolutionary advantage to that?
I mean, it seems to be. Well, all we have to do is just think about it a little bit. So, we know that ions are very useful to firing things, right? We've talked about the anionic basis, and sometimes we need to integrate signals. So can you integrate if it's a gap junction?
Well, it would be kind of like the bits in the computer would be ones and zeros, right? It's on or off, I would figure.
Yeah, but you can't really integrate. The second cell just automatically fires because there's nothing to slow it down necessarily, right? So there's just a natural...
Oh, okay, I see what you're saying. Oh, so it doesn't act as...
It's not so much of an integrator as our canonical model.
It passes the action potential right on through. It doesn't even stop. It just goes through the dendrites, soma, right on through the...
Remember I said it's so fast.
Okay.
Like it's a nearly...
Okay, I guess I was figuring that...
So what you can do is you can take... If this is your electrical recordings of your two neurons, and you just record them, and you see the trace of them, and the electrical trace is nearly identical. There's no delay. It just whoop, slides right through.
Do you find those specifically in certain neuron sets?
Yeah.
Do all neurons have them?
No, they don't. And that's why I said it was under that little section that I titled exceptions to the rule, or like weird exceptions to the rule, or something like that.
Okay, so in some circumstances, it's a one continuous.
Yeah, and so it's just... It's like one of those, like, it's kind of like retrograde signaling. It's like, it's a weird exception to the rule. It's not the rule.
Gotcha. Okay.
Traditionally, it works in presynaptic to postsynaptic for signaling. Very occasionally in a weird exception, they do nitrous oxide or carbon monoxide. That does the, retrograde signaling. Same thing with the gap junctions. It's not the primary way because, obviously, the chemical transmission is the primary mechanism trying to...
Okay, that makes a lot more sense.
Yeah, and so... so to your broader question, what might be the advantage? Well, we need to integrate signals. I mean,
and you can't integrate if it's just one, because there'd be too much electricity going all over the place. It'd be one constant signal. Everything would fire at once.
Yeah, so you have a yellow light at a stoplight. What do you do?
Speed up.
What do you do?
Speed up or slow down. Yeah.
Well, or slow down. Well, you need to do one. You can't do both, right? You can't be like, I mean, you ultimately have to do one.
Right.
How do you
slow down?
How do you know what to do?
I guess, well, the yellow light, I know that tells me that a red light is about to come up. So I slow down. I'm not sure I understand.
But sometimes you speed up.
If I think I can make the red light.
Oh, so you have to integrate some other information. So it's not just about the light. Now you start looking around.
Now it's environment.
Yeah, where's the safety factors? Is there cars coming? How far away am I? How fast am I going? Is there alternative traffic? Right, you have to integrate all this other information.
Okay.
Gap junctions aren't so good for that.
They just blast right on through.
Think about, we talked about spatial summations, temporal summation, those principles of how neurons are integrating both excitatory and inhibitory information coming in.
Excitatory. The logic functions kind of are vanished because the decision's already made with the presynaptic neuron. The action potential goes through the gap junction, right on through. There's no decision making involved.
Not in the context that we're referring to decision making. A philosopher might disagree, and I'm not a philosopher, so I'm trying to be very very precise in how I try to characterize things. But yeah, I know what you're saying. And you're right. Yes, there's not a lot of decision making going on in the gap junction with our traditional.
I guess algebraic decision making is what I was trying to say. Trying to find the algebraic sum. You can't.
Integration is a more neutral way to say.
I see what you're saying.
We don't want to appropriate consciousness to the cells that they're making decisions.
Right, exactly.
But they're integrating.
Okay, that makes sense.
But I mean, conversation. Yeah, I mean, I know exactly what you're saying.
Thank you, sir.
Yeah, totally.
Hi, Dr. Addante. I just
Howdy.
wanted to say, I know you saw me going up and down. I don't want you to think I was ditching. I just had to go to the bathroom. I didn't realize it was so loud.
I was judging you the whole time.
I know.
I was totally judging you.
I was like, I feel bad. You probably think I'm really in class, but I'm
Come on, man. Would I do that?
I know. I'm sorry. I'm sorry. Thanks.
It's all good. Look, you've taken this class out, what, 4 times? 3 times?
3.
Look, the fact that you even still show up is impressive to me, so you're good. Yeah, don't sweat that stuff. Yeah. Alrighty, guys.
Hey, just a quick, well, hopefully it's a quick question. I heard a long time ago that when people take like SSRIs, how it, as you explained, traps it in there and kind of causes it to work more, but it's not necessarily actually producing more serotonin, it's just utilizing that serotonin more.
Mm-hmm.
And I heard a long time ago that because of the way that process works, it teaches the brain that it doesn't necessarily have to produce more serotonin. So when you stop taking the drugs, that you go back into the diminished amount or even possibly less serotonin than before you were on the drugs. Is that actually accurate?
Anecdotally, I would not be surprised if that makes a lot of sense.
Okay.
I'm not a neurochemist at the level of specificity, but yeah, I mean, that makes a lot of sense, and I've heard similar things like that.
Okay.
But and we haven't talked about it a lot because we're only just scratching the surface on this stuff as a very basic introduction. But one of the things I like to dial in, particularly in this class, I tend to do it at the very end of the semester when we talk more about the lecture on drugs and abuse and mechanisms of action that underlie patterns of addiction and drug abuse. But the principle that I think is reflected by that is that I usually try to tell people think of this system, right? So we talk a lot about this both
in like my workbench,
electrical and chemical balance, really, right? And how these cells are, indeed our brain is operating. And I think of it as like this normal, healthy neurochemical soup. Right? Because it is a soup.
Right, yeah.
It's fluid. It's comprised of stuff and, chemicals. And for the most part, we think of things probabilistically and like, the bell curve of humanity. We all have a fairly normal, healthy balance of that neurochemical soup, for the most part.
Right.
Now, most of us end up talking biologically and scientifically, we all have a rough average of a healthy neurochemical soup for a certain point in time until we all start screwing it up by introducing these external ligands. That healthy neurochemical soup is pretty screwed up on the morning of a hangover after a long bender. We've all been hungover. Right? We've screwed up that healthy neurochemical soup. Now, fortunately, in that situation, it kind of clears out, and it has a relatively benign long-term effect if it's only a short-term method, essentially, right? We clear it out, we recover, and that healthy balanced soup can help to rebalance itself. And you've talked about a few of those mechanisms, the uptake and clearing that out. But when you start messing with things in a more substantive way, like introducing, medicines. I mean, SSRIs are medicines, as we consider them medicines, but medicines is a subjective term. They're just drugs. Some drugs are legal, some drugs are not. My drug of choice is...
Some drugs we recommend.
Yeah, well, like SSRIs. And I mean, my drug of choice is caffeine. And I'm probably a full-blown addict by any definition of the word. And I can absolutely attest to the fact that it messes you up. Man, when I crash off of it, I crash. And it is not fun.
Yeah, some people get shakes in the morning if they don't have it too early or at the right time that their body is conditioned to take it.
For energy drinks.
Oh, don't even get me started. I told you, I told you worked in the nightclub scene a while back, and we started. Nobody didn't even know what Red Bull was. We used to get 12 free cases a week just to do promos and give them out. We didn't even know that we were making Red Bull slushies.
They used to make one called
That's how old I am.
They used to make one called cocaine. It's illegal now.
What's that?
They used to make one called cocaine that looks like Red Bull. Same can. It's red, but if you rip off the outer label, there's different ingredients and it's crazy.
Yeah, no, it's legit.
It tastes like cocaine. Like it's crazy.
Yeah, we'll talk about that.
I was gonna say sharing stories here.
Yeah, I mean, like that's a whole other thing. I'm happy to chat about it, but I mean, in the context of your question, if you think about like we have this healthy, normal, natural neurochemical soup, and then we do mess with it. But so does our natural environment does anyways in some regards, and that's more broadly epigenetics and things like that. But we start really intentionally chemically messing with things, be it drugs that we call medicine or drugs that we call legal, like caffeine or alcohol or nicotine, or drugs that we call illicit and illegal, cocaine and heroin, all these other things. Especially the ones that tend to be the ones that we call illegal are the ones that tend to create and wreak the most havoc on this normal, healthy, balanced soup that prevents it from really resetting that normal, healthy balance. And so when you talk about that in the context of your question of the SSRIs, and then it leads to differential regulation in the production of these chemicals, because you're confusing it.
It's almost like you're teaching it. Well, okay, so what we made is now balancing and it's sustaining here, and we don't need to produce more in order to have the same effect. And I hear it diminishes the production use. And I don't know if that changes after you get off the drug for a while or not.
Well, it's a really difficult thing to test and investigate. And people work on it, and we find certain clues, and we work on it. But, I mean, it's difficult to even address things like that experimentally. And, of course, we have ideas and models and things that seem to capture that well. But a broader issue that I think you're tapping into is this idea that, for the most part, most of our pharmacology and medicine treats symptoms, not problems. Right?
Yeah,
so we're treating a symptom of lowered serotonin. Well, here's a really nice way that we can bump up serotonin. Great. And a lot of people, you know, it's really done wonders for them. It does great things. But what we've done is we've treated a symptom, not necessarily the underlying mechanism of action for which the problem is actually served. And so when we treat that symptom, it makes all the sense in the world that there might be a side effect or some other biological correlate or consequence that changes as a function of that. Because our system is designed to adapt and change. Remember we talked about upregulation and downregulation of those receptors. Just like you said, it taps into that same idea. When you start messing with our healthy neurochemical soup, it can be really difficult to reset it to that natural way. I swear, every weekend I start the next week thinking that I'm really going to stop the caffeine. I'm going to move from coffee to tea, and every now and then.
There's still tea. There's still caffeine in tea.
Dude, I'm working.
I'm just letting you know. I just wanted, you know, break the tea gently.
Well, some teas
it's still there.
Some teas don't.
Okay.
And those are the teas I tend to prefer. But I also like to prefer the ones that do have caffeine as a moderated step down on the way to it. And I tell you, it's a brutal couple days. But it's great when I can dial into it, and then something comes up, and I end up getting tricked back on the thing, and. But it's really hard to get back to that healthy, natural neurochemical soup, because it's really stick with that analogy. It's a soup. The waves are moving all over, right? And they're all moving in this direction. And then you stop, and it's like, oh, and you try to go in the other direction. Now there's not that systemic sort of whirlpool pattern. Now it's all out of whack and not systemic. It's a little chaotic for a little while until you can get that natural, normal flow of those waves.
Right.
In a very general level of conversation. Does that, I mean,
it makes sense.
Did I answer your question?
Sort of. I mean, you also answered it in the way of that we're not really sure totally, but at the same time.
How could I answer
probably. I mean, it was the question as posed was kind of technically a yes or no question, which you can't just answer it in a yes or no question.
I mean, I could, but I might be lying.
Right.
I mean, I like to be very scientific about it. You know,
for the way you answered it, it sounds like the way that I believed it to be is correct. Is like it does, because it does. It messes with you, but we don't know that after that you stop taking the drugs, if it will eventually recorrect itself. That's dependent on other factors.
Depends on the drug.
Right.
Like I said, I mean, you recover from a hangover.
Yeah.
You know, you don't recover from Korsakoff syndrome, which is a consequence of alcoholism. You know, so it's just, I guess, the question itself is very broad, and so it necessitates a very broad answer. Unfortunately.
No, it's okay. No,
and I can give you a specific one. A lot of people like really concrete yes and no's up and down, but what they get is people who ultimately end up lying to them in terms of a detailed fact. And I try to be very scientific about things.
I like your answer better than just a yes or no anyway.
Because
I think I understand it a little bit better.
The way that most science works, right, philosophically, is that what we can say, or what we do say, is limited by what we can say. And what we can say is only based upon the available evidence at hand.
Right.
And we can make a probabilistic guess.
Right.
Now, most of the stuff we get on medical textbooks, science books, I mean, there are things that the evidence is so strong for that we have an incredibly good reason to make a very good probabilistic guess. We're saying, yes, this works this way, this way, this way.
Right.
But that's why I introduced at the very beginning of class, science is a process, not a destination. So even though it looks like all the available evidence says, yes, this is the thing, it still makes it very difficult to say anything for absolute certainty.
Right, because we're still learning.
Because there's so much more to discover. And it might be that there's this other nuance that works in this way or that way. And so I'm just scientific.
In 100 years, they're going to go, everything they learned was the things that were wrong.
Well, didn't I say that in the class about this?
Saying, yeah.
The first class, there was like a lecture about.
They believed this and they're so silly.
Calculus is like, everything you know about math is a lie.
Well, you know, but I mean, like, that's not bad. It's, you know, I mean.
No,
I hope all of my papers are the exceptions. No, I'm kidding. But, like, you know what I mean? Like, that's, you hope that, first of all, I hope that people prove my stuff wrong on a personal level, because that means that I did something that was cool enough that somebody cared about to say.
Challenge it.
Yeah. And so, like, I mean, like, I really believe in that model, in that ethos of, like, that being okay. Like, because that's awesome. That's how it's supposed to work, right? And so you'll find a lot of scientists, they get married to their work or to their theories or their models. Sometimes.
That's why I'll never be married.
Sometimes. Are you married to your models and theories and hypotheses?
Yeah. Well, I'm a workaholic.
But that's different.
Anything I do.
But that's different. You can work at your marriage and be a workaholic at it, right? Aha, yeah. You didn't think about that, did you? No, but you know what I mean. So I've always just, when you get married to your theories and your own models, you get in really dangerous waters, because then you can be taken to places where you, in retrospect, people will look and you're like at you as being you might sometimes have acted a little silly because of your love for your own model and theory, and that's not scientific.
Right.
Scientific should be agnostic and blind of whatever the available evidence shifts a preponderance towards.
Well, and it's like, I mean, like the majority of things that we've learned or experimented or figured out is all still technically theory.
Oh, yeah.
Because it's not concrete, and so that's like you're saying, that's the dangerous of like.
Have you ever heard,
this is completely right. Well,
have you ever heard of Darwin's law of evolution?
Yeah, I can't quote it.
You've heard of Darwin's law of evolution?
I've heard of Darwin's law of evolution. Yes.
Really?
Why are you looking at me like?
It's called Darwin's theory of evolution.
Oh, well, I'm sorry. I wasn't thinking of the words.
See?
No.
So it's just an example I try to use in class, because you said everything is just theory.
Yeah,
like, and that's a big one that people generally accept. And they're like, yeah, this is kind of the way that we think things work. We don't call it
the law because it's
even Newton's laws. There was one I read a couple years ago. I'm not a physicist, so I'm a bad person to ask. But somebody said that there was a challenge to one of these weird little nuances of one of his laws. I mean, laws are tricky. There's not many of them for good reason. Like we are very conservative and as a body of science of saying this is our theory of how we think things work. And hopefully we can all remain open-minded enough so that we can embrace the challenge to certain theories.
Well, and also you want to be able to open yourself up to the possibility of change because you want to continue to make sure that you're learning more too.
Indeed. And that's why I say sometimes some scientists can get taken in some pretty dark waters if they start to get married to their theories and their models too much.
Because you shut yourself out.
And then people look at that as being foolish later on, and hindsight being 20/20, they're like, oh, that guy walked into the kind of mess over there because wasn't really being as open-minded as he could have been.
Right.
And that happens. I mean, look, we're also human, so that happens to all of us, right?
Yeah. Ego and pride, no.
Never. I've never been accused of this. But you know what I mean.
Yeah.
And but it's funny. So the last paper that I had written about some memory finding that I had, and it was just last year, so it's in my mind, and they had done a press release here at UTD about it, and like the last line in the paper that they quoted me on was something about like. Like I hope, like I said something like, I don't even know if this is right, but I hope somebody tries to prove it wrong.
Nice.
Like, I do. Like, I don't know. It's just one finding. It might be wrong.
A,
That's okay.
that means I'm interesting and my work was interesting.
And it's still a citation.
And B, it means that somebody else is interested.
If it's cited, it's still counted as a citation. They don't count pro and minus citations. They just count citations. Yeah, see what I'm talking about? Right? So you throw a little challenge out there.
I watched everyone was laughing.
So there was a professor who I had lunch with at one point when I was a postdoc, and he was a real famous researcher on the amygdala, and he had said that he had a paper that he wrote about the amygdala that challenge or did something or another, and it wasn't very big of a thing, and it challenged some stuff of some famous lady's work. And then she wrote a paper where she made a comment in it, or a commentary about his finding and about how he was full of crap and it was a terrible finding, or
Had like a beef.
yeah, you know, whatever.
Yeah.
I mean, which is fine. This is how science works, right?
Yeah.
You look at something one way, I look at it another, and you say, I think it's this, and you're like, no, that's crap. I think it's this. Now we have a framework from which to test a hypothesis, which is fine. Right. I mean, Golgi and Cahal, right? They had that beef. Like,
Right.
Nothing wrong with that, right? We learned a lot from it, and so she did that, and he was telling the story, and he's like, yeah, he's like, she brutalized me in that paper, and it was the everlasting best thing that ever happened in my career. He's like, nobody knew about me before that paper, nobody knew about the work, and because she basically bashed or challenged it, you know, however you want to make it sound a little bit more eloquent or whatnot, but he's like, because she did that, it brought so much attention to that paper that everybody else started citing it now. And
Nice.
so, I mean, there's, you know, it's not a bad thing.
Plus, it's always good to have the excitement of a challenge.
Yeah.
Or
It's a controversy.
competition.
I mean, science is kind of like pro wrestling, right? I mean, first of all, everybody loves to hate somebody. Like you gotta love, and the villains are more popular than the protagonists, right? So like, you know, everybody loves Stone Cold Steve Austin and The Rock when they were the villains at times, but everybody also loves the hero of the Hulk Hogan and all that, right? And,
kind of like 80s wrestling.
well, I don't know wrestling outside of the 80s. I did amateur like American wrestling after that, so I only.
Do you have a story for everything?
Yes, I do. The trick isn't getting me to tell the story. The trick is getting me to shut up.
I feel the same way.
Yeah. So